Welcome, everyone. Thank you for coming to the School of Psychology's annual winter seminar. For those, I'm Lauren Brent, by the way. I'm in the Center for Research in Animal Behavior in our School of Psychology, and I have the role of welcoming you and giving you a bit of information about the event before we kick things off. So those of you that have been in psychology a while will know that every year we have a winter seminar. It's a chance for us to celebrate the end of the coming of first term, the coming of the winter holidays, and all the festivities associated with that. And typically this is an internal speaker that we have. So we have somebody, one of our own, tell us a bit about their research, and then afterwards we have a few drinks, a bit to eat, we mingle, it's all jolly and nice. This year is extra special, however, because, of course, one of our own has been promoted to professor, and so in addition to the typical winter celebration, we're also celebrating him and his achievements. So the structure will be basically similar to what you might expect. We'll have a talk. We will not have questions after the talk, just so everybody knows, in keeping with the celebratory tone of the event. So we'll go straight out into you're not allowed to grill anybody. So we'll go straight out into the foyer of the forum for some drinks and some nibbles. Everyone is welcome to join us there. And we'll also have a few presentations and toasts, so please do join us for that. Tonight is also different because you might notice quite a few unfamiliar faces, non-psychology people in the audience. So we have people from other schools at Exeter. We have some of the executive group from CLES here. And we have some people who've come from all over the UK to join in the celebration with us tonight. So please also welcome them. And yeah, thanks for all of them for coming tonight. And with that, I'll move on to one of those special guests who has the wonderful task of introducing our guest speaker. So Professor Mike Kent is a professor of evolutionary biology in biosciences in our Center for Ecology and Conservation down at the Penryn campus. And over to you, Mike.
So, welcome everyone. I'm really delighted to have been asked to introduce Darren on this auspicious occasion, his inaugural professorial lecture. I'm a relative newcomer in Darren's life. However, I have been working really very closely with him for about 5 or 6 years, developing and testing theoretical ideas about the evolution of life histories, which we'll probably hear about, I think. A little bit later. But during that time of working really closely with Darren, I've developed an enormous respect and admiration for him, both as a person and as a scientist. So it's a great joy to be able to celebrate his achievements here in this way. I've come up on the train from Cornwall, bearing numerous messages from your friends and colleagues down at the other campus. There's another inaugural tonight in Cornwall, which was a bit of a clash, but they send their messages of warmth and congratulations with them. Now, I asked permission from Darren if I could kind of paint a little bit of his backstory, because I think it's safe to say he's had a rather non-standard route to his new elevated position of Professor of Animal Behavior. So Darren grew up in Scarborough in North Yorkshire, not so far away from my hometown of York. And at school, I think it's safe to say that school wasn't a massive success for you, Darren? No? Because as some of you know, Darren is quite severely dyslexic, though this was undiagnosed at the time, and I am imagining, having gone through the same schools, that North Yorkshire schools in the 1980s probably weren't the most forgiving environment or the most supportive or understanding at that time for pupils with dyslexia. So the careers teacher said to Darren, okay, here's my advice, there's 3 jobs for you. You can be a farmer, a fireman, or a carpenter. So, of course, Darren chose to be a farmer. And he actually left school, and Julie went and milked sheep for a year in the Yorkshire Dales, and then went on to agricultural college just outside of York. So a life as a farmer was looking like it was beckoning and everything was well laid out. But in between milking sheep, Darren was reading The Selfish Gene by Richard Dawkins. And like many of us, he became hooked and inspired by the evolutionary concepts that are in that book. So by the time he got to Leeds University, and he was diagnosed with dyslexia and was able to use computers to communicate much more effectively, he encountered modules on ecology and evolution and really decided to ditch the idea of being a farmer and instead become a behavioral ecologist. And luckily for us, his talent was recognized by a very renowned behavioral ecologist at Leeds, Professor Jens Krause, who took Darren under his wing and as a supervisor of his undergraduate project and his PhD and a postdoc, he really nurtured Darren's great talents. So Darren then went on to a lectureship at Bangor and joined Exeter in 2008 as a lecturer here, and has had a fantastically successful career since then. So there's lots of things I like about this story, but a couple of them are, I think, that there's many different routes to a successful career in science. And the other one is, I suppose, that people's inspiration and passion can come at different ages, at different stages of their lives, and in the most unlikely of places, such as in a milking shed in this, on this occasion. So since then, since 2008, Darren's been very successful at winning funding, at publishing great papers, and has established a really world-renowned reputation for the quality of his science. And I think, having worked with him, he's got many qualities. I want to pick out a couple of them. I think, and maybe people who are here who work with Darren also will recognize this, that he has just an incredible natural curiosity and tenacity, and a very good eye for finding really good problems and puzzles, and won't rest until he's either solved it or at least illuminated it for other people to try and attack and solve. And the second quality I think I've been incredibly impressed by is the way that he makes all of those who work with him, students and colleagues and collaborators, he makes them feel valued and their opinion is respected. And this is something I've really stood back and admired, and I've tried to emulate it to try and imbue my students and colleagues with a similar sense of value and the feeling that what they have to say is worthwhile, because it's an amazing talent he has for putting teams together and getting them not only to work together, but to do their best work together. So, enough of the embarrassing intro. I want you to join me in giving a very warm welcome to our speaker for tonight, Professor Darren Croft.
Okay, can you all hear me at the back as well? Yeah, brilliant. So, thank you, Mike. Very kind words, and it really has been great to work with you over the last 5 or 6 years, and I look forward to many more years of working together. And in putting this talk together this evening, I really spent quite a lot of time in trying to decide what to tell you, whether or not to give you this history of a little bit of everything that I've touched and worked on, and I will do a little bit of that, or to tell you about one particular story in detail that I find absolutely fascinating. I decided to go for the latter. And I want to tell you about this project that started around 10 years ago and has really grown into a fantastic research question and project and a team of people working on it. So I'll just check that this works. So the talk structure, what I want to cover, just to give you some idea of how long you have to wait before we get outside to drink some wine. There's 3 steps to this talk. The first one, I just want to extend a little bit on what Mike said and tell you a little bit about my journey from PhD student to here in Exeter, and starting out at Leeds working on these little fish, and how that then led to working on these big fish. And I know they're not fish, but they are called blackfish by the locals. And then I want to focus in on one particular area which I find absolutely fascinating, and it's leading on to new questions, and the work is generating new questions that I hope in 10 years' time we're going to have equally exciting science to tell you about. And I also want then to tell you a little bit about the future and some of the new things that we're working on at the moment. And you've already seen this next slide. But I thought, wouldn't it be nice, because I'm giving a talk about one particular, I wanted to acknowledge everyone who I've worked with and published with. And I had this brilliant idea in the morning that it wouldn't be great to put a slide together with all my co-authors. And I thought, that would be great, and that'll half an hour's work or something. I did a little search online, I realised I'd published with 142 different people, and it actually took me a whole afternoon to put a slide together with all of my co-authors, and I just want to reassure you, I haven't actually published with guppies or cows or badgers. They were just placeholders where I couldn't actually find a photograph of somebody. And so I want to say thank you to all of these people, and these people have been my mentors, and certainly Jens, Mike mentioned earlier on, they've been my friends and collaborators. And I've had great enjoyment of working with these people, and many of whom are in the audience. One of the things that was really enjoyable about putting this slide together was many of the people on this slide I published with as undergraduate students or as MSc students 10 years ago, 9 years ago, and actually going on and looking at where they were today gave me a great sense of achievement to see just how successful many of these people have gone on to be. So thank you. My research, if I was to give a general overview of my research interests, I'm interested in groups that form these dynamic fission-fusion societies, so where animals join and leave groups. The groups are not stable, they sort of rephrase that, they can be stable, but they're not exclusive, so animals can come and leave groups. They join, they leave, and this typifies actually group living across taxa, so insects, ungulates, fish, and all the way down to the resident killer whales, which we will talk about in a bit more detail. Even in human societies, we form these fission-fusion groups where we join and leave different groups, and group membership is fluid. And there's really three questions that I guess have kept me awake at night sometimes, got me excited when I wake up in the morning at others. So how are these societies structured? What are the mechanisms and constraints that underpin these structures? So how does the behaviour of individuals drive the structure of these groups of animals? And the bit that I'm going to tell you more about is what the consequences of this structure are for ecology and evolution, and we're going to talk about life history evolution in the killer whales. But before I do that, I want to tell you a little bit about where I started that journey that led us towards these killer whales. But these are the three questions that have really captured my imagination over the last 10 years or so. And that really started, if I can play this video, we'll see a flock of starlings here being predated upon by a peregrine falcon. And you'll notice that these animals are coordinating their behaviour, and they're moving as one cohesive group, and they may be a little small on the screen, but if you keep an eye, you'll see this peregrine dash through the middle, and the starlings will evade it. And actually the peregrine doesn't successfully capture a starling in this footage. And that's pretty incredible. These birds are aggregating together, and by doing so, they're able to confuse the predator, so the predator isn't able to catch a starling. And if it does manage to catch one, they dilute the risk of any one of them being captured. If I was a predator and I came into this lecture hall, and I could only take one individual, the chance of any one of you being captured would reduce as a function of the number of animals or the number of individuals in the group. So animal groups are incredibly good at avoiding, or individuals in groups are incredibly good at avoiding predators. And we also know that the animals in the middle of the groups have lower risk of predation than those at the edge of the groups. And this was really the starting point when I was a PhD student. We had really good information on the benefits that individuals could get from being in a group, but did these groups just consist of individuals that were interchangeable between groups? Or was there some structure within those groups that's important for how those groups function and avoiding those predators? And I was fortunate enough to convince Jens at the time that he should let me go to Trinidad and work on these fish that just happen to live in the rainforest and see Trinidad here off the coast of Venezuela. And it wasn't just because the fish are in Trinidad. This is a model system in ecology and evolution, and a really great system for studying how prey have evolved to avoid predators. And in this northern mountain range, and Dick James, who I'm very pleased is here, was key in helping me guide me through this research. In the northern mountain range of Trinidad, there are these barrier waterfalls, and if I play this video, you'll get to see some of them. This is in the Taruri River, taken earlier this year when we were out on a collection trip. And the guppies have been able to get above these waterfalls, but the predators haven't. This particular waterfall is too big, and actually a curious biologist in the 50s decided there are no guppies there, so we're going to move some there. We'll take a bucket and tip some in there, and actually that turned out to be one of the best experiments to illustrate evolution in the wild, because these fish have evolved for the last half a century under different ecological conditions. So we're able to use this system to be able to look at animals that have evolved under different predation risks. And a lot of the work that I've done on these fish has actually been looking at these guppies in these habitats and how they've evolved to avoid predators and the social structures that have evolved to allow them to do that. And so as a young PhD student, I went out to Trinidad and I spent a lot of time by the side of these streams. It's a fantastic place to work. The water's crystal clear. And you can watch the fish and you can watch them interacting in the river. And the dynamics, I wanted a dynamic society to work with, and they joined and left groups on a timescale of seconds. So I found the system I was looking for, but it was a little too dynamic for what I was really, how am I going to study these relationships, these associations, this social structure? And so that led us on to thinking about how we can track individuals through time and their social interactions through time. So we were tagging these little fish by, under a binocular microscope, injecting a small amount of paint, a latex under their epidermis, and with different combinations and different colours, and then releasing the fish back into the streams, we're able to construct these social networks of these fish. And you'll see these in a few points through the talk, so I'll just tell you what they are. So a node is an individual, a dot is an individual, and a line, an edge, means that these two animals have interacted. And by doing this, we could start to look at the structure of the social interactions through time and build up a picture of how these guppies joined and left groups over days, over minutes, weeks, months even. And in doing that, we found that these fish actually have quite stable groupings, and these females in particular form these really stable structures. So we're taking time series data and visualizing it using these networks. And that allowed us to describe the structure of these societies and go on to ask a whole lot of questions that I could tell you about, but I'm not going to tell you about. But it was really the starting point for thinking about how animal societies for me are structured over time, and then what does this structure mean for the individuals within those structures. So do these females do better than the females on the outside? The females here are the white nodes, the males are the red nodes. And that led us to realizing that this, obviously we hadn't created a new methodology here. People have been doing this in human societies since the 30s, but actually as behavioral ecologists or people interested in animals, we hadn't really thought or synthesized the ways in which we can use this data to construct these graphs and then analyze these graphs. And so we brought that information together with Dick and with Jens and with Dan, who's also in the audience, in a monograph and then in an edited text. And that has propelled many studies for myself and hopefully for others as well. And I've been very fortunate to work on a wide range of study systems through my career, even going back to my roots as a farmer. We've been working with cows and sheep here in Devon and trying to understand how their groups are structured and the consequence of that for their health and welfare and their productivity. And throughout this research, the questions have allowed us, this methodology has allowed us to link the individual to the group to the population, but then look at the consequence of the population structure for the individual, the group structure for the individual and the population structure for the individual. So really to think about how individuals can drive these group structures and the consequence of the population structure or the group structure for the individual in the group. And that was really the starting point, and that's the crux of the research I want to tell you about with the killer whales. I left the University of Leeds in 2006 and took up a lectureship Bangor in North Wales, and at this point I'd spent my time on a farm in the Yorkshire Dales and in Trinidad studying fish, and I arrived at Leeds and they offered, not Leeds, Bangor, they offered me a job, and the first thing the head of school said to me, "Fantastic, Darren, you're now going to teach a course in marine mammal biology." So it came as a bit of a shock, but it turned out to be the start of an incredible journey, and I met Emma, who's also in the audience over here. And Emma told me about a study system that she was incredibly passionate about, which was the southern resident killer whales. And Emma had spent a number of years out there as a volunteer and really recognised the potential of this data, which had been collected, we'll see in the later slides, over a long time period, to be analysed and formally analysed using the techniques that we'd been using on the guppies. And so Emma undertook her MSc dissertation on the southern resident killer whales. And just to give you an indication of where they are, this is my office here on a Google Earth image. And if I just click this, we'll zoom out and we'll go on a little journey over to the Pacific Northwest of the USA. Killer whales are widely distributed across the globe, but this population is particularly unusual in that it's a resident population of whales, and it occupies the Salish Sea region. The Centre for Whale Research here is on the west coast of San Juan Island. And the whales actually, the Centre for Research is the building that's close to the water there, and the whales actually swim past this side of the rock here, right past that building, and that's provided this organisation with an incredible opportunity to be able to document these animals year on year, and I'll tell you a little bit more about that as we progress through the slides. But this really started with Emma and her master's project. I moved to Exeter, as Mike said, in 2008, and I was fortunate that Emma moved across to be with me, having initially taken up her PhD at Leeds with Jens, and in the end moved down to do her PhD here at Exeter. And this project really blossomed and grew into quite a significant international research project involving researchers in 3 countries, involving a non-profit charity, and it's been funded by both NERC and the BBSRC. And I feel incredibly lucky to have worked with these people. And when I say I actually mean they, later on in the talk. These are the guys that have done the work, especially the PhD students and postdocs that are on the bottom of the screen here. And it's great to see Lauren have gone on and successfully fledged from this project and have introduced this talk. Lauren worked on this project as a postdoc a few years ago. So these killer whales are very unusual, but we know probably more about this population of killer whales than most of the marine mammals in the world. And that's because they have this very unusual strategy and very specialised foraging strategy where they feed predominantly on salmon and predominantly on one species of salmon. So 85% of their diet is Chinook salmon. There are different ecotypes of killer whales throughout the world. Many of them feed on marine mammals, and you'll have seen the fantastic footage that David Attenborough will have shown of killer whales predating on blue whales or grey whales. These whales are slightly, they're clearly very intelligent animals, but they're very restricted in their diet. So they will kill harbour porpoise, for example, which is perfectly good protein, but they won't eat it. There's something about that's culturally conserved that they will only eat this salmon. It's absolutely incredible. There's 2 populations that occupy this area of the Pacific Northwest. The northern residents are in the northern region of British Columbia, or around British Columbia, the northern end of Vancouver Island, and the southern residents around the bottom of Vancouver Island there. Another interesting fact about these whales is they're the same species, but they don't actually, there's been no gene flow between these 2 populations for thousands of years. So they're culturally conserved to only one species of fish, which actually probably will end up being their demise and leading to their extinction. And also, they don't mate with the neighbouring population, which just seems, from an evolution perspective, a really bad idea, but fascinating for us. What's sad about this population is that SeaWorld in the 60s and 70s managed to convince the US government that there were thousands of these animals and there was a sustainable harvest that could be taken by removing these animals, which were then worth very lucrative public display. Killer whales went from being this feared creature, this killer creature of the ocean, to something that could be tamed and commercialized in aquariums. And through the 60s into the 70s, SeaWorld actually removed or killed, the marine parks removed or killed 66 whales from this population, leaving just over 70 whales left. And the population has never actually recovered from that. And many of the whales that were captured died in captivity and had a much shorter lifespan than be expected in the wild. This captures the pressure on there was pressure put on the US government in order to assess the stability and the viability of this population for harvesting. And Ken Balcombe in the 70s had just finished on another project and was employed by the US government to go out and count the whales. And methods had been developed by Dr. Michael Bigg, where he'd realised actually that all of these animals have a unique fingerprint. Their saddle patch, which is this area near the dorsal fin, this white area, actually is different between individuals, and by photographing these animals, they're able to tell them apart and they're able to identify individuals. And in 1976, Ken went out and he started off photographing these animals, and he's been doing it every year, more or less every day that those whales come into that region. Ken has gone out and photographed them for the last 43 years. And there's a picture of us together in 2016, and he was out doing it again this year. And this is what Emma had recognised as having incredible scientific value, this long-term, with the guppies we were watching these fish for 7 days, perhaps 30 days. These animals have been watched for 4 decades, recording their births and lives. And using the same methodology, we've been able to then take that data and describe those patterns of associations. And these killer whales form a very tight society where different family groups or different matrolines will come together. And this is the type of video footage and the photography or the videography that the centre's been able to get. And you can see in this video, you can clearly see the dorsal fins that have notches or different shapes. And you can also see those saddle patches that have allowed those individuals to be identified. And from that type of data, if we take that on days and across weeks and across months and across years, we can start to build up a picture of their society where, again, individuals and nodes and their social interactions are represented by these edges. And I'll tell you a little bit more about their family structure on a later slide. I want to emphasize just how incredible this data set is. And if we take probably the most famous of these whales, which died was pronounced dead in January last year, J2, or as the locals call her, Granny, and we'll see why she's called Granny. We don't know her date of birth. Her date of birth is estimated to be around 1911 at the earliest, but it could have been 20 years later than this. And that's because the study didn't start until the 70s, so her birth date was estimated based on her size, the size of her offspring, and whether or not she was reproducing. So there's some error around her age estimate, but as we'll see, it doesn't affect what happens later on in her life. But just to put some milestones in time here, to give you some context, the Titanic sank a year after she may have been born, if we take her earlier birth date there. We have World War I, then World War II. She was definitely alive by the start of World War II. Sometime in the 50s or 60s, she went through menopause, and I'll tell you a little bit more about that as we go on into later slides. But she had her last calf in the 50s, 1950s or 60s. And we don't know the exact time because actually there was nobody recording those whales at that time. One of the most famous captures for taking whales into captivity happened in 1917. She lived through that, so she was there during the Pen Cove captures, and there's actually still a whale in captivity that was captured during those captures. In 1976, then, Ken Balcombe was commissioned and employed by the US government to undertake the survey, and that marks the start of our long-term survey. This is me in 1976. And I show that because I want to give you a real insight here into just this is science across generations of scientists. It's not just science across generations of animals. This study has been going as long as I've been alive. In 2009, this is J2 in her 70s, possibly in her 90s, breaching there. This is, I reproduced during the time that J2 didn't reproduce, so remember she went through menopause here and she didn't have any offspring. Well, I managed to have some offspring, and I managed to get them to see J2, so they met her in 2016. And I find this picture fascinating, and I'm sorry you're not going to be able to see the detail in this picture, but you'll have to take my word for it. J2 is on the right-hand side here, and just about, I don't know, if you were looking at a clock, it'd be, 11:58. There's a salmon. A really small line there in the picture. If you don't believe me, come down and look at my computer screen after the talk. And J2 is actually catching that fish. So remember here, she's aged somewhere around 80, definitely, possibly over 100. She's catching that fish to feed it to J45, this male who's just lost his mother. So she's not reproduced here in certainly 40 years, possibly longer, yet she's still catching fish to feed to other members of her matriline. And I just find this storyline absolutely fascinating, and it really highlights just how unique this long-term study is. That these individual animals have been followed through 4 decades. And the other thing that this highlights is actually Ken's mission was to go for a full lifetime of an individual, and it's only actually halfway through, although he's been doing it his whole lifetime. If I was sat in my office down in Washington Singer and I thought, I know what, I want to study menopause in killer whales, I would be 84 by the time I had the data to be able to do this. It really is just incredible. And unfortunately, she was declared dead in January 2017. So one of the things that this long-term data has highlighted that I want to emphasize is this really unusual pattern of reproduction. That these females have this period in their, they will have their first calf when they're in early teenagers, and they will have their last calf in their 30s, 40s, but have this incredible period of life when they're post-reproductive. And from an evolutionary perspective, that's really puzzling. It really is a challenge that if we think about evolution and natural selection, it's honed individuals to increase the number of offspring that they pass on, the genes passing on to the next generation. And the easiest way to do this is directly by having more offspring. Why should an animal switch off reproduction partway through life? These animals, and this is a key to answering this question, have a really unusual social structure that they share with only one other species, these short-fin pilot whales. And neither males or females disperse from the family group. So sons and daughters live with their mother her entire life in this matriline, in this stable matriline. In birds and mammals, normally one sex disperses. In most mammals, males disperse. In most birds, females disperse. But there's normally always a dispersing sex. These are the only 2 species that we know for sure where both offspring never leave the family group. They stay. And that's actually key, and it will become key later on in the talk. But menopause is really just, or the reproductive aging is really just part of aging. And so I thought it was important just to kind of give a quick overview of why, the current thinking for why we age. Why do we age in the first place? Because reproductive age is just part of the aging process. And in the early part of the last century, it was thought that actually selection happened at a species level, and animals were pre-programmed to die, so we didn't compete. It was good for individuals to die, because if we kept living, we would overpopulate, and there would be just too much competition, so there was pre-programmed death. And actually, that got pushed to one side in the 50s, when Peter Medwar was actually giving his inaugural lecture at University College London in the 50s, and his lecture was entitled An Unsolved Problem in Biology. And the unsolved problem in biology was aging. It was, why do we age? And what he realised was that actually there's always more young individuals in a population than there are old individuals. So many individuals will die because of starvation or predation or disease before they get to be old. And so typically this is an age structure for this, elephant population in Kenya. And one thing to note here is that the age bins actually jump from 5 to 10 once you get to 25. So there are even fewer older individuals than the graph makes it appear. And so what this means is that there are few animals actually get to be old, so that there's stronger selection on young animals simply because there are more of them. And because of this, mutations can accumulate in the population. And so it really is this, that because there are fewer old individuals, there is stronger selection on younger individuals. And George Williams, a few years later, realised actually that this can lead to something else, that strong selection in early life can have detrimental effects in late life, because if there's a gene that provides an advantage in early life, then because there are more individuals that will express that gene and gain that advantage, but most individuals will die before they ever experience the deleterious effects of that, then selection can actually favour traits in early life that have a cost in late life. And this is one of the mechanisms that we think is responsible for many of the diseases that we experience now as we age, this antagonistic pleiotropic selection. And George Williams so committed to the study of senescence that he actually documented his own senescence. And he recorded his own running speed through his 50s and 60s for running around a park every year. And you can see this decline here. But the key thing about aging is we would expect the body to age, just generally the body to age. Our hearing starts to go a bit fuzzy, our eyesight deteriorates, we get a bit slower when we run, our muscles start to waste. But we expect the body to decay, the different parts of the body to decay or age at a similar rate. And so we expect the reproductive system of the animal to age at a similar rate to the rest of the body. And so if we look at this plot here, and we'll see a couple of these in the next slide. This is age along the X, or along this axis here, and this is the probability of survival in red, or fecundity in blue. And so the higher this red line, the more individuals are alive. The lower the red line, more individuals have died. The higher the blue line, the more individuals are reproducing, or reproducing with a higher rate. The lower the blue line, the fewer individuals in the population are reproducing. And this is generally what we expect to see, that the reproductive system and the rest of the body age at a similar rate. So animals maintain the ability to reproduce throughout their life. These two toothed whales really buck the trend in that, and this is what we saw before in that graph, in that their survival extends long before, or long after rather, their reproductive period. And this is really unusual, but just to highlight how unusual that is, I'm going to show you a few of the figures. So we're going to see these again, so just to emphasise, the blue graph here, animals reproducing. So we see the killer whales here start reproducing in their teens, hit a peak in their 20s to 30s, and by 40 that was flatlined, no individuals are reproducing. But the red line is still positive, so there's still individuals alive in the population. And this is just a plot for females. If we look at hunter-gatherer societies in humans, then we see, that's the top right figure there, we see a very similar pattern whereby there's a peak in facility in midlife, but the survival extends well beyond the age at last reproduction. And indeed, we'd argue there's been convergent evolution, so that the reproductive life history of the killer whales and the humans have clearly evolved independently, but they've evolved to look very similar. And strikingly different to all other primates, as far as we're aware as well. So if we just click on and look at some of these figures, I could have picked any 4 species of non-human primates, and they all pretty much look the same, whereby the blue line extends over the red line throughout life. So these animals maintain their ability to reproduce until the end of life. And as far as we're aware, it's only humans, killer whales, and these short-finned pilot whales that have this unusual life history, that have this termination of reproduction partway through life. And that really presents a puzzle for evolutionary biology to understand how that could have evolved. There are other species that will live after they've stopped reproducing. So, for example, if we keep animals in captivity, there's some lab rats there, we can prevent them dying from predators and disease. We can put them on breeding cycles that will exhaust their supply of eggs far beyond that they would experience in the wild. So we can create animals that have still alive but have stopped reproduction in the lab, but it's very artificial. Some animals have this suicidal reproductive strategy where they just have one shot at reproduction, such as these Pacific salmon, and then they die. And they will live for a short period of time after they've reproduced. But from an evolutionary perspective, it's not adaptive, it's just a consequence of getting to that point, staying alive for long enough, and then they'll just live for a little bit. They get carried over for a little bit. There are some examples of where old females get pushed out and are no longer able to breed. So in the Seychelles warbler, young females dispersing into the territory push the female out, and she can no longer reproduce. She helps, she becomes a grandmother, but she maintains the ability to reproduce. And something very similar happens in carpenter bees, but it's the daughters that push the old females out. There's only one other species, as far as we're aware, that actually terminate reproduction, and that termination is adaptive, and it's an incredible species. And it's an aphid. And this tiny little aphid produces a gall, so it simulates the tree to grow like an abscess, and then it has its offspring inside of there. And as those offspring start to mature and develop, it moves to the outside of the gall. It turns its body into it reabsorbs its reproductive organs, it turns its body into a glue bomb, basically. And those little things sticking out on the end of the aphid there are actually balls of glue. It sits on the outside, on the opening where its offspring are going to emerge, and it sticks itself to predators. This is a ladybird larva. And so we could argue that this is, I mean, it's got menopausal life history, but we're going to put that to one side and focus on these three species. The aphids are slightly unusual in that they have clonal reproduction, so they're related by one to their offspring. But it's an incredible bit of natural history. There's been an incredible interest over the last 50 years as to why menopause has evolved in humans, from both an evolutionary perspective, but also a great interest from a medical perspective. And clearly, over the last century, there's been advances in modern medicine, which we've all benefited from, or are benefiting with our lifespans. Since medieval times, the mean lifespan at birth would have been around 20 to 30. Lifespans have tripled since then, with people regularly living into their 80s now. And so one possible reason for menopause in humans is simply it's just an artifact of this increase in longevity, that actually our ancestors never experienced this trait because they never lived long enough to get to that point in their life. The reality is, however, that when we look at these mortality rates, it's childhood mortality. So it's mortality of children that's high, and that pushes down, if we think about the mean age of longevity. And actually, and unfortunately, a really good example of this can be found where I started my PhD on the island of Trinidad. And in the early 1800s, Trinidad had been captured by England from the Spanish, and the population of the land rather was fertile and cloaked in rainforest. And the UK wanted to farm Trinidad and farm its rich and fertile lands. And the way at the time this was being done is through moving slaves from the west coast of Africa to the Americas, and the slave population was clearing the plantations of Trinidad. And around that time there was a great pressure on the UK Parliament to abolish the slave trade, and one of the things that they did, the abolition movement, was they were able to push to get a register of slaves on the island of Trinidad. So every slave by law had to be documented. And that paints a very sad and very disturbing picture of the slave trade and for the slave population. Around 40% of slaves actually died within a 3-year period. The mean life expectancy for a slave at birth was only 7 years. And without replacement, the population would have gone extinct. So they would have been lost completely from the island. And that led to this movement of millions of people, actually, from West Africa over to the Americas. But this record, just like we will see with the killer whales, allows us to go back and look at life history patterns, because old births were recorded as well. And in this population, if children survived that critical period early in life, actually women could expect to live around 1/3 of their life post-reproductive. So even under the harshest conditions of mortality in living history outside of war, menopause is happening. So it's not simply an artifact of modern life, of our longevity. This is something that's deep-rooted in our evolutionary history. And for humans, there's a key hypothesis as to why this has evolved, which is that it's due to the benefits that grandmothers can gain by helping their offspring reproduce and their grand-offspring survive. And here we have a nice example of that with the Hadza of Tanzania here. and they dig for tubers, and tubers, you can see, dig for them with a stick. It's manual physical work, and then these tubers need to be broken down in order to be cooked. And again, that's hard labour. And actually the grandmothers can play a really valuable role in society, in the family group, by helping their offspring to get food and their grand offspring to survive. But why do they stop reproduction? So they can benefit, but why do they stop? And I'll leave that question there because we'll come back to it. But in humans, it was long thought that the costs of late life childbirth, that actually there are risks of giving birth late in life, and that those were sufficient to explain why you should stop, and then switch over to helping. As we'll see later on in the slides, actually, those don't appear to be sufficient. But we start with the grandmother hypothesis, the idea that old females can help their offspring survive. And that was our starting point then for looking at this in these killer whales. And these killer whales have a very, as I said, a very unusual social structure whereby the sons stay, so they live in these matrilines, and the sons stay in the family group, but they don't mate with their sisters. They go to another group to reproduce. But they return to their mothers. So what happens is when a son reproduces, another family group, another matriline rears the calf. The son returns to his mother. When a daughter reproduces, the calf is reared within the family group. And so just after I arrived in Exeter, actually before Mike and I had had a chance to really meet, Mike had been thinking about this in the context of menopause, the evolution of menopause in humans, and predicted that mothers should preferentially, in killer whales, mothers should preferentially support their sons rather than their daughters. And the reason for this is if a son reproduces, the group carries no cost for rearing the calf. Whereas if a daughter reproduces, it's another mouth to feed. Females need about 40% more salmon if they're lactating. We know salmon is a really critical key indicator of mortality for this population. Struggle to find enough fish. Having another mouth to feed could actually lead to mortality in the group. Also, sons have a higher reproductive success. They can mate with many females. If a female has a calf, she's not going to have another one for 4 years. Whereas a son could mate with a different female every day and have many calves in a year. And so that was really a starting point for Mike and I working together and testing some of these ideas with these killer whales. And what's just incredible about this population, and I can't emphasize this enough, is just how strong that social bond is between mother and son. And you'll have a 30-year-old male, not just in the same bay as his mother, or in the same area, but swimming within 3 meters, clinging to the side of his mother. And they do this throughout their life. And so, I'd like to thank Malila for producing this little diagram for me. And so, we can use the same analysis that we would use for insurance companies would use if they're analysing our hazard of dying. If I phone up the insurance company and I say, okay, I want some insurance, they will ask me how old I am, whether or not I'm male or female, if I smoke, whether or not I have other high risk factors, and from that they will determine or predict how long I'm expected to live for. And from that they will then calculate how much I should pay them in order to get life insurance. Well, these survival models we can use to make predictions about mortality risk for animals if we know their history. And so by using the same type of analysis that insurance companies would use, we can actually start to ask questions about what influences the survival and mortality of these animals by using this long-term data set. And so when Emma came down to Exeter doing her PhD, I very clearly remember the conversation in the PhD office with Emma, and we had just got talking, and somehow Emma said to me, well, these males are all just mummy's boys. What do you mean they're mummy's boys? Well, they die when their mummies die. They do what? Okay, we have to test that. We have to interrogate that. And this had been seen in the community of people watching these whales, that when a mother dies, the son tends to die as well. And actually, what we're able to do is bring a team of people together, both with the data and with the analysis techniques, to be able to test that hypothesis. And the results were really striking. So males don't live as long as females. Males have a shorter life. So you're going to see a few of these graphs. So this is time since birth, so this is age along this axis here. And in this axis here, it's probability of survival. So at the top, at 1, everyone's alive. And as this line starts to drop, it means individuals are dying. And so the steeper that slope of decline, the quicker those individuals are dying. So the black are males and the red are females. And you can see that the males are dying quicker than the females. And so this gives us a measure of survival. But we can ask about events within an individual's life. Again, using this analysis that can then determine their longevity or their life expectancy. So what happens if the mother dies? If the mother dies and the offspring is aged 15, and by then they've reached sexual maturity, then males actually, you can see the line drops further. So their hazard of dying increases. So these young males at 15 years old, if their mother dies, then they're more likely to die as well. What was really incredible about this analysis is what happens for old adult males. And what's interesting about this population is these old males that have a higher reproductive success. And if a mother dies when the offspring is aged 35, the hazard for the male increases around 8 times. So he's 8 times more likely to die in the year after his death. These old sons are dependent on their mothers for survival, and to some extent, the females as well, but only to a little degree. It really is the sons that are dependent on their mothers. And this was really exciting, because this is what Mike had predicted a few years earlier, that these old females should be investing in their sons rather than their daughters. How do females help? And that was really the next question for us. So we'd shown that these old females are providing these benefits, but how can they be providing benefits to their group? And one idea that we tested was that they may act as repositories for knowledge of when and where to find food. And so these old individuals have experienced many years of trying to forage for salmon. And these salmon are migratory, I'll get there in the end. So they're moving into the rivers to spawn after spending 4 or so years at sea. And there are a number of spawning events across the years, or a number of migrations within a year rather. And it's incredibly important that these whales time it right to hit the right river or the right bay where the river's flowing out into, where they can collect and catch the salmon. And that differs for different rivers and different years. And if we just look at a plot here, we can just see variation in salmon number across years, but this also varies within a year. And remember, these whales are preferring to feed on these big, juicy Chinook salmon, and generally neglecting or ignoring the other species that they could be eating. And so one hypothesis was that these females could be importing. Emma, I don't know how we convinced you to do this, Emma, but you watched 751 hours of video of whales doing this. And Emma recorded who was who, so able to quantify patterns of leadership by looking at who was out in front of the group and who was following. And from that, Emma left the project after doing that. And Lauren took over the data set and the analysis. And we were able then to actually ask who was leading the group and who's following the group across these years. And again, we can come back to this network. Now it's a leadership network, whereby individuals are nodes again. The size of the node is the age of the animal. Here they're coloured by sex. And an arrow means that this individual followed this individual. So we can look at who's got lots of arrows coming into them, they're the leaders. Who's got lots of arrows going to other individuals, they're the followers. And if we look at this, we can see, it's clicking forward a little bit too fast. We can ask, okay, so who leads? Do males lead more than females, or females lead more than males? And we can see here that females are actually leading the group more often than males. What's interesting is if we compare females to reproductive and post-reproductive, it's the post-reproductive females that are doing the leadership. So it's not just females leading the group, it's the old females. They're having their last calf in their 30s to 40s. And from that point forward, they're really occupying this front position in the group, and they're leading the group around these foraging grounds to find the salmon.
And this is really important because in years of low salmon, if you think back to that figure that was wobbly with the low years, we know that in those years whales die. So they really struggle to find enough food. So the knowledge of where to find that food is really important for the survival of their group. And just going back to looking at who follows, we see that males are following their mothers much more than daughters are. And this is quite interesting. So if I think about my ability to find my way around a city if I followed somebody, I'm useless. So if I was to walk to a new city and so somebody says, we'll go to this restaurant and we're talking and I'm just following, and I have to find my way back to the hotel, without this, I've got absolutely no chance because I've not had to pay attention to my surroundings. Whereas females from a very early age are out on their own somewhat more and building up this spatial map that's allowing them to navigate. And perhaps that's partly one reason why they're able to buffer the loss of their mother more than males. This Chinook salmon, unfortunately for the whales, is also commercially a very important species. And that means that there's, in addition to fantastic data on the whales, there's also incredible data on the abundance of the fish. So we can ask about how the prey abundance affects the leadership patterns. And this is just an index of salmon abundance here along the bottom, and then a leadership score in the vertical for post-reproductive and reproductive females. And just the thing to note here is that one line is flat, and that's reproductive females. It doesn't matter how much salmon there is, reproductive females generally lead about the same rates. Whereas when salmon is in low supply, that's when these post-reproductive females really kick into this leadership role, and they're then guiding their groups around these foraging grounds when we know that it's critical for their survival. But it doesn't tell us why they stop reproduction. So we know that elephants, for example, leadership is really important in these elephant societies. These old matriarchs have information on how to deal with lions, how to respond to predators effectively. They also have information on other social groupings, the potential for conflict between groups and how to resolve that conflict. We also know that they're important in keeping their grand offspring alive. Yet a 60-year-old female will still be reproductive. So, you know, if we were to look at the ovaries of a 60-year-old adult female elephant, she'll probably still have about 11,000 eggs. So still a significant amount of reproduction left in her. They don't undergo termination of reproduction. So we've shown that these old females provide benefits to their offspring and to their family group, but not why they stop. And surely a female could continue to have a calf every now and again and still be a leader of the group, just in the same way as these elephants do. So there's really something missing from this story, from this description of why menopause has evolved. And we can come back to this unusual family structure, this unusual group structure that the resident killer whales have. And under these family dynamics, what actually happens is that relatedness changes as individuals age. And this is something that Mike and Rufus realised back in 2008 for human populations, and that the same thing applied here with the killer whales. On the graph on the left here, there are 3 lines. And what this graph is showing is how related an individual is to its local family at different points in its life. And as we move this way on the graph with age, animals are getting older. The red line is its relatedness to females, the blue line is its relatedness to males, and the black line is its average or relatedness on average, taking the local relatedness of both males and females into account. We can just talk through this and it will help to understand the graph. So a calf is born into a group. Obviously its mother is in the group, it's born into the group. So it has a high relatedness to females because its mother is there. So the red line is high at birth, so the red line stays high. The female then starts to reproduce when it hits to be a teenager, and its own calves, its own female calves, stay with her. And so it stays high, so the red line stays flat. Females have the same relatedness locally to other females in the group throughout their life. What's really interesting here is what happens with a local relatedness to males, because a calf is born into a group, but the father was from a different matriline. So the father isn't part of the group. So at birth, Females have a very low relatedness to other males in the group because the father is absent from the group. But what happens as she ages is that her calves and her male calves stay with her and stay in her group. And that actually means that her relatedness, her local relatedness, increases as she gets older. And we can see that blue line, age here 1, 2, 3, it doesn't mean they're 1 year, 2 years and 3 years old. It's just an indicator of different points in the animal's life, 3 being old and 0 being young. As the female's aging, she's getting more related to males, and the consequence of that is that her relatedness on average increases. And this was a fascinating prediction, because in most mammal societies you see the opposite pattern, that relatedness decreases as individuals get older. We're able to test this using this long-term data, and if we just pay attention to this part of the graph here, because this model didn't have menopause in it, and this is the point of menopause, there's really a good fit here between the theory and the empirical data. So relatedness to females stays constant, relatedness to males increases to the point of menopause, and the mean relatedness to the group increases as these females are aging. This sets the scene for some dynamics in the family which can be characterised as conflict. It's not just about helping, there's also a dark side to this as well. It's about conflict in the family. And so Mike and Rufus had formalised this as the reproductive conflict hypothesis. And we were able to extend this and test this in these resident killer whales. And what this means is that if a mother and daughter breed at the same time, so if a mother has a calf at the same time with her daughter, they're competing for limited resources needed for reproduction, like salmon or babysitters, access to help. But because of the shape of this graph, theory predicts they shouldn't compete equally, because young females are less related to their group, so they should fight harder to get those resources. Old females are more related, so they should show more restraint and not compete as hard. They should be more cooperative, less willing to harm the group, less willing to take that extra salmon away from another individual. Remember that picture that I said where you had to believe me there was a salmon there? There was an old female who later died 2 months later, catching a salmon to feed to another individual. There is a strong selection to help in late life and harm in early life. And we're able, working with Rufus and Mike, to actually develop this into a simulation, a model, to predict what old and young females should do. So because of this asymmetry in young females being less related to their local group than old females, young females, this side of the graph here, should invest more in competitive efforts than old females, and as a result they should have higher fecundity, because they're investing more in getting access to salmon, or not giving salmon away, or not babysitting, so actually they should have a higher reproductive success. Let's get back a couple. This actually happens a number of times, quite a few times in that long-term data set, that mothers and daughters breed at the same time, so we're able to go and look at what happens to the survival of these calves when these females from different generations, so you've got the old generation, the mother here, she has a daughter, and then her daughter has a calf at the same time as the mother, so that these two females are competing, or their offspring are competing at the same time. And if we do that, the results are really striking, and they fit with this prediction that we saw from the model. So again, we're looking at survivorship here going on the vertical axis and age here on the horizontal. As we start to move down, it means animals are dying, they're less likely to survive. We can see the offspring from the younger mother, so that's this offspring here in green, and this offspring here in purple. The offspring from the older female has a much lower chance of surviving if these calves are born into this competition. When we look at the cases where they're not in competition, the calves actually don't differ in their survival probability. It's only when these two females from different generations reproduce at the same time, that the calves from the older females actually suffer and have a much higher risk of dying, and dying in early life. And this is wasted effort. If a female reproduces and the calf dies, then actually this is wasted effort. And what these females are doing then is switching from a strategy of trying to reproduce to a strategy of helping. And this is partly because they're pushed out of their reproductive seat, if you like, by their daughters, who are competing more with them to gain access to the limited resources needed for reproduction. And we think something very similar happened early in human evolution, and that the relatedness patterns of ancestral human societies, for different reasons that I won't explain, or patterns of dispersal, different patterns of dispersal, look very similar in that local relatedness increases as females get older. And we can talk more about that later on. But just to conclude then, so post-reproductive females provide significant survival benefits to their offspring. We showed that by looking at what happens when these old females die. But one of the ways they do this is by being these repositories for knowledge of where to find food and sharing that information by leading the group around these foraging grounds. But the menopause has evolved due to both the benefits that these old females can give, but also the costs and the conflict in the family over reproducing. And that these young females actually are investing more in harming and in getting access to that limited food needed to reproduce than these older females. And these older females then switch over to a helping role. And we think something very similar happened in early human evolution. Unfortunately, this population doesn't seem to have a very bright future, and it really balances on the knife edge. There are only 76 individuals left. It's a closed population, so there's no dispersal from this population. There's no new gene flow into this population either. And there's less than 30 reproductive females in the population. Actually, a recent paper that we're involved in predicts that they're likely to go extinct within the next 100 years. It only actually needs one oil spill or one disaster, and the whales will go extinct. And even if the current threats just remain the same, which clearly they're not going to, the population will flatline. Any further decrease in salmon, and this population's on a downward spiral. The Centre for Whale Research, who would be incredibly fortunate to work with, continue to fight for these animals and a voice on behalf of these animals. And I just want to say, if you're wondering what to get someone for Christmas, membership of the Centre for Whale Research is a fantastic Christmas present to give your loved ones. But seriously, if you want to learn more about these whales, there's an incredible amount of information on this website. I just want to finish just a couple of slides of where we're going in the future, because that was about things we'd already done. Our next puzzle that we're really excited about is captured in this graph, which is these males are dying, and they're dying long before females. And we tend to think about this, males kind of banging heads with each other to get access to females, and that means that they suffer from disease or they run out of energy, they're more likely to be eaten by predators. That's not happening here. These males are living with the same family group as these females, yet they're dying in their 30s, whereas the females are living into their 80s, potentially longer. So one of the things we really want to understand, our next challenge, our next question, is understanding why the lifespans differ between these two sexes. And just a paper just published, Sam has been working on, just published last month, shows, again, these are survival plots, but looking at how central these males are. The left graph is males, the right graph are females. Males are much more sensitive to their social environment. So a male that's less central, that has less connections to his group, is more likely to die. Females, they're unaffected. It doesn't matter how embedded, how connected they are to their family group. And we think this social structure is really the key to unraveling why these sexes differ in their survival. And that's something we're hopefully going to be working on. One of the ways we hope to do this is by getting an aerial view of these animals. So everything we've talked about so far has come from these shots from a boat or video from a boat of looking at the animals from the side and recording their dorsal fins. Technology now means we can fly these relatively low-cost drones over the whales and start to get this incredible photographs and video footage of actually animals interacting and who's sharing food with whom. Next summer we're going to be flying our drone over the whales and recording behaviour. The types of behaviour you're going to be able to record, you're not going to hear the sound in this video, but captured in this video. So this is a predation event. This is a killer whale capturing a salmon. And this was, we're on the boat. The salmon decided to use the boat as a refuge. So this salmon about this size, probably about where Mike is over there, charged towards the boat. The boat's not very big. The killer whale in close pursuit swam under the boat, and then circled round and round like this. And this is a wide-angle lens. This animal's probably only about 5 meters off the boat, just looks a long way. Catches the salmon, and then another whale comes in from the side and they share the food. We're able to see a lot of this, but we miss a lot of what's going on by looking at the side. By looking above, we're going to be able to really unravel the social lives of these animals in a way that's just been prohibitive before now. We know very little about vast numbers of marine mammals that live in the ocean. And one potential way into this, actually, unfortunately, many of these animals strand, and occasionally strand in large numbers. We're looking at using this data to try and plot the reproductive lives and the total lifespan of other species by making use of data from strandings. I just want to finish where we started by thanking all the amazing people I've had the opportunity to work with, and just highlighting the team that have been key in this work that I presented on the killer whales, many of whom have come to the room today, to Exeter, to support this talk, which I'm incredibly touched and honoured by. And also the funders who have funded my research throughout my career. And you all for coming. Thank you very much.
I'll use that here. Right, so now for the final bit. I'm not going to keep you very long from your drinks and refreshments outside. For those of you who don't know me, my name is Dan Charman. I'm Pro-Vice Chancellor for Life and Environmental Sciences. And one of the great pleasures I have is to come to these events and thank some people, actually. I mean, that's the most important thing. Also to listen to the lectures, of course, which are always fascinating, just as in this case. So there's two groups of people I want to thank, and then an individual, fairly obviously, that I want to thank, but I'll come to him in just a minute. So the first thing is these things don't happen by accident. There are people involved in making sure we get all the arrangements right and so on. And thanks today, particularly to Lauren and Karen. I can't see where Karen is, somewhere here. For Karen in the psychology office for organising this event. Thank you very much, because it's really important that we get it right and have lots of people coming. Secondly, thank you to all of you in the audience, because these are fantastic celebratory events, you know, and I think it's important, you know, people only do one inaugural. So if Darren, not that I'm suggesting you should do this, moves to another university, he will not do another inaugural. This is a one-time thing. This is when you become a professor, you do this. So this is a really fantastic celebratory event, and it's celebrated with all of you here. So thank you for coming. Finally, Darren, you know, what makes a good inaugural lecture? I think it's a mixture of stuff, and I think Darren's got it all in there. I think it's a very difficult thing to do a good inaugural. We're all used to talking to our peers in science and research, and you know, we can pitch it a certain way, and we expect a bit of criticism back for it, and so on as well. But actually, for inaugural, you've got a whole mixture of people here. Some of the people know a lot about the subject. Many will know nothing, just as in my case, actually, really, I knew nothing about the subject. So you've got to pitch it right and make it entertaining. There's got to be something in it for everybody. It's got to have some pictures. It's got some pictures of people, because, you know, it's a celebration of career as much as anything else. And all of us in our careers encounter other people. I think Darren did that absolutely fantastically with that screen full of people. Amazing that there were only a few people he couldn't actually find photos of, really. And it's also got to have photos of family, and it's definitely had photos of family, which is great to see, and thank you too. I mean, it's great to see you here, and you know, I hope you realise how clever your daddy is now. You know, that all these people have come to listen to him and hear him talk about his work. So great to see those photos, and of course the inevitable. Only one photo of you as a child, actually, Darren. I'm quite impressed that you managed to restrain yourself to those childhood photos. So thank you for giving us such a brilliant lecture. I won't keep you any longer from refreshments, but let's finish with a round of applause for Darren.